the organizers for this opportunity, and this is a very complex title. We have to cover cancer stem cells and metabolic reprogramming and the metastasis. But the focus of the presentation today is to understand the link between the metabolic reprogramming and the cancer stem cells and the metastasis. So the background for those who are not familiar with the cancer stem cells, the cancer stem cells differ from typical stem cells in two of the major properties. One is they are non-responsive or uncontrolled, they are non-responsive to any regulatory mechanisms, and that's why they are uncontrolled. And they also have a limited ability to undergo differentiation. In the literature, you can find several synonymous terms interchangeably used instead of cancer stem cells. Some of them are like tumor-initiating cells, and some of them are dormant cancer cells, but they all refer to the same property, which is the stemness, the cancer stemness. The original model or hypothesis for cancer was that all tumors are heterogeneous, and each cell in a tumor can proliferate, and all these cells are, in essence, have the tumorigenic potential. But based on the understanding of the cancer stem cell theory, now we know, of course, all tumors are heterogeneous, and all cells in a tumor can proliferate, but only a subset of cells have the tumorigenic potential, and those subset of cells are referred as cancer stem cells because they exhibit the markers of stem cells. So this has been initially debated, and then later on several laboratories have documented evidences to prove this existence of cancer stem cells or the tumor initiating cells. And what is shown here is a nice experiment where lung cancer cells were isolated from a patient, and then they were fractionated into two categories based on their resistance to therapy, the conventional chemotherapy, and based on the expression of the markers, the stem cell markers. And both of them, when compared for their tumorigenic potency or resistance or the stem cell phenotype, both of them were identical. And that proved only a small population of cells in the entire lung cancer had this capacity, and they all resemble stem cells in the cancer. So these cancer stem cells have been reported or have been investigated in many types of cancers. This is just a short list. And you have the cancer stem cell population in almost all malignancies, including rare malignancies. And now we will just switch gears to the metabolic reprogramming. So the metabolic reprogramming in essence means rewiring of metabolic circuitry, which is otherwise called as tumor metabolism. Because this is so specific or unique to cancer cells, often they are referred as cancer metabolism or tumor metabolism. So in cancer, all these major biosynthetic pathways or the metabolic pathways which utilize glucose and glutamine, or glutamine and the glucose are rewired or altered, and that is why this metabolic reprogramming is so significant to cancer growth. So the most popular among all these alterations is the glucose metabolism. And as you all know, in the normal tissue, in the absence of oxygen, glucose is converted into lactate, and that is pumped out, and this is called anaerobic glycolysis. But in the presence of oxygen, it will undergo mitochondrial oxidation, where the glucose is converted into ATP, and it produces more ATP molecules. However, in cancer, you will see, even irrespective of the presence or absence of oxygen, the glucose is primarily converted into lactate, and this phenomenon has been reported by Warburg long ago as the major biochemical hallmark of cancer cells. And even though this property, this biochemical signature you can call, was reported more than 90 years ago, it was not until the last decade recognized as one of the hallmarks of cancer. In 2011, in an excellent review article where Drs. Weinberg and Anahan listed the hallmarks of cancer, they included, this is the metabolic reprogramming, which is called as deregulation of cellular energetics, as one of the hallmarks of cancer. And this metabolic reprogramming or altered energy metabolism has been seen in metastatic cancer cells. And So far, what we knew until the emergence of the metabolic reprogramming was oncogenes drive tumorigenesis, and the tumor suppressors try to counteract and they try to suppress the tumor. But with the emergence of the knowledge with the understanding of the metabolic reprogramming, now we know oncogenes actually upregulate metabolic reprogramming, or they facilitate metabolic reprogramming, and that is critical for tumorigenesis. And tumors under unfavorable conditions, they also initiate metabolic reprogramming to sustain their continued growth. So now we will look at what is the connection between cancer stem cells and the metabolic reprogramming. What do we know so far is the cancer stem cell markers can be induced by hypoxia. Hypoxia is a low oxygenation condition, and the cancer stem cells contribute for metastasis. So we know the cancer stem cell markers are upregulated by hypoxia, and they are present in they facilitate metastatic process. From a metabolic reprogramming point of view, we know metastatic cells exhibit the altered metabolism, which is a metabolic reprogramming. What is not known so far is in the metastatic process, during the early steps of metastasis, what is the role of the CSC phenotype, which is the cancer stem cell phenotype, or the metabolic reprogramming. So we wanted to investigate this. Before we get into the details, we need to know what are the early stages or early steps of metastasis. So in any metastatic process, yes, cancer cell from its primary tumor site has to get released from its attached site, typically epithelial origin, and they enter into blood vascular system, and once they identify or recognize appropriate niche or the microenvironment, they escape from the blood vascular system and then form the new tumor in the secondary site. So this involves three major events. One, anaikis resistance. Anaikis is nothing but a process of cell death, especially for cells which are dependent on attachment for their growth. Once they are released, they have to undergo apoptosis because that is not their survival mechanism. But in cancer, these cells, even though they get released, they don't undergo anaikis, and they have a resistance to anaikis. The second stage is the epithelial mesenchymal transition, because these are epithelial cells. In order to get transported through the circulatory system, they have to assume a phenotype or get into a different phenotype, which is a mesenchymal phenotype, and this is called epithelial mesenchymal transition. Not only that, once they reach their target site, they have to reverse the phenotype, which is a mesenchymal epithelial transition, so that they can grow into a new tumor at the secondary site. So the idea of investigating metastasis has to include anaikis resistance and epithelial mesenchymal transition, if possible, mesenchymal epithelial transition as well. So the current models for So this is the same point that I was going. So the current in vitro models that mimic metastasis that is used in research laboratories for metastasis, the oldest model is the Boyden chamber design, which represents one of the salient features of metastasis, which is invasion. And this is an excellent model to study local invasion, but you don't see the anchorage independent growth, which is nothing but assuming or assumption of the EMT, which is epithelial mesenchymal transition. And also we don't have any proof or evidence to show in the local invasion whether they require the cancer stemness or the tumorigenic potential. It could be just a local invasive nature. So the second type model, which is very recent and probably not very recent, probably 3 or 4 decades old, the 3D spheroid model, where people started culturing the cells in the presence of matrix, which is extracellular matrix, or popularly known as matrigel, and the cells will be mixed and they'll form aggregation and form a spheroidal structure, which is a 3D multicellular spheroid structure. This is an excellent model because it mimics in vivo tumor in a realistic, in a real clinical situation. But the drawbacks of this 3D model is they do not select cells which are more aggressive, and there is no selection. The matrigel aggregates the cells together, and then there is no proof for the reversal of the EMT. We know there is epithelial mesenchymal transition because the cells become circular and spheroid, but there is no necessity or there is no opportunity to reverse the circular cells in the spheroid into to mimic the epithelial phenotype. And finally, there are a few concerns about the impact of the complexes that are used in the matrigel or the extracellular matrix on the biology of cancer cells itself. So we wanted to develop a model, an in vitro model, which can recapitulate all the features of desirable steps of the metastatic process, which is I'll go through this first, which is including the EMT, MET, and the anchorage inhibitor growth. This is a conventional monolayer model, and this is the 3D spheroid model that I discussed above, and this is the model that we have right now. Oh, sorry. There is something missing here. Okay. All right. So here, what we do is, from a parental monolayer, you culture those cells under a dish which has ultra low attachment. There is no matrigel, there is no extracellular matrix, and there is no other agent that is mixed with the cells. You just plate these cells as regular on these plates, but these plates are coated with the hydrogel, which prevents the attachment of the proteins onto the substrate or the substratum, which is the base. So the cells would not attach at the base, and they tend to be floating. And once they float, only the cells which has the potency to grow under anchorage independence, they aggregate and then they form the spheroid structure. And then you transfer these spheroids. You don't have to trypsinize or release these cells. Just transfer these spheroids and place them back on a similar culture, regular culture dish, and they tend to spread. And this is shown here. This is the parental monolayer, and if you culture them under this condition, you see the formation of the spheroids, and this is a higher magnification of the same spheroid. And once you see the spheroids and place them back into the regular culture dish, they tend to migrate, and you can see a downhill pattern. There will be a leading edge of migrating cells, followed by the other cells which are climbing down from the spheroidal structure. So if you give a few days, like 3 days or 6 days, depending upon the tumor type, you can see the complete reversal of these spheroid cells into a monolayer. So what is the advantage here? There is no ECM or matrigel mixing, and they do show anchorage independent growth, which is the EMT phenotype that you see here. And then they do integrate the reversal of the EMT into mesenchymal epithelial transition during your reversal. And more importantly, I don't know why I'm not saying here, that more importantly, they select the cells. Here, you don't make all the cells to become spheroids. You can see, actually, I can show you in the PowerPoint later, you can see there will be dead cells, and only the cells which are resistant for anchorage independent growth, they get into the spheroidal structure. So having established this model, we wanted to verify if this is really a good in vitro model. So we did a qPCR and looked for the markers of cancer stem cellness, and the EMT, which is epithelial mesenchymal transition, and the drug resistance, which is a common property of cancer stem cells, and the metastatic signature, which is invasion. All these markers showed an increase. If you see the red one, that refers to the reverse to monolayer, which means the anchorage independent cells after the reversal into monolayer, and this is a parental monolayer in blue. And in all these markers were upregulated in the reverse to monolayer. So this means maybe we selected the cells which have the potency or the tumorigenic potential, or maybe these cells also have upregulation during the process of selection. It could be either way, but anyway, what we have is representative of very aggressive phenotype based on the qPCR. But to achieve functional effect, we need to confirm if this is phenotypically valid. So what we did, we then investigated by immunofluorescence microscopy whether the EMT marker vimentin is upregulated in the reverse to monolayer, and as you can see, there is a upregulation of the vimentin, which is an indication of the EMT marker in the reverse spheroid monolayer compared to the parental monolayer. The blue refers to the nucleus, and you have the expression in the parental monolayer, only limited expression, whereas in the reverse to monolayer, the expression is upregulated. They are so diffused, you hardly see the blue DAPI staining. And then we also looked for the transgene, which is similar to TWIST, another metastatic protein, which helps in the metastatic process. And we tested in multiple cell lines, which is here, these two are human hepatocellular carcinoma cell lines. This is a breast cancer cell line. In all these cell lines, the upper panel refers to the parental population or the heterogeneous population, whereas the bottom panel refers to the reverse spheroid monolayer. You can see a significant increase in the fluorescent intensity, which indicates the upregulation of the marker transgene. And then we wanted to confirm if the CSE markers, which is the cancer stem cell markers, if they are also upregulated. And then we chose CXCR4, EpCAM, and CD133, the prominent markers of stem cells, cancer stem cells, and those are upregulated in the bottom layer. You have wherever you have the RS, it's the reverse spheroid. You have upregulation compared to the parental population, and that is true for all the three stem cell markers. And here again, we tested HU7, MCF7, and ESCAPE1. I'm just showing a representative. We actually tested them in 5 different cell lines, and in all of them, the selection process confirmed you are isolating cells which are positive for cancer stem cell markers. And then we checked one of the properties of the cancer stem cells is the chemoresistance, and we used a very known glycolytic inhibitor. Since I used this inhibitor, I just tested, but we have also done extensive investigation with other drug combinations. And in all of them, the red line indicates the cells that we isolated based on the in vitro model, and the blue line refers to the parental population. And you can see there is an increase in the IC50, the dose requirement to achieve the IC50, even in a time-dependent fashion. This is true, and we confirmed the marker, which is ABCG2, a marker which is responsible for the drug efflux. And if this marker is upregulated, you can confirm that will be chemoresistance. And we see in the RS or reverse spheroid population, there is an upregulation of the ABCG2 compared to the parental population. And this is true in different cell lines. This is FG2, MCF7, and HU7. All these 3 cell lines, and then in the other cell lines also, the data support the same trend. And then we wanted to investigate if the isolated population shows any difference in the invasive ability or in the metastatic process. So this is a parental population. You can see the purple cells which are migrated, very few in the parental population, meaning you have a very small proportion of invasive cells in the parental population, whereas in the in vitro model that we isolated, you have a large number of cells which show the invasive invasion. And this has been supported by MMP2, very popular proteases, metalloproteases, one of the proteases which helps in the invasion process. And you can see if you compare to the parental, there is an increase in the expression in the reverse spheroid monolayer, and this is true with all the other cell lines that we tested. The functional verification was done by Zymogram, and even the Zymogel, you can see an increase in the activity of the MMP. And then we wanted to verify in the animal model. See, if you inject a small number, we injected here only the least number of cells possible, which is 10 to the power of 5 cells, and within 4 days, you can see in the reverse spheroid cells, you can see the appearance of multiple nodules, and here you hardly see a comparative. I mean, you can see there is only one animal which showed tumor development at the site of injection. So these were injected intraperitoneally, and the cells were actually stably expressed with the luciferase gene, so that you can image them with the bioluminescent imaging. And I'm showing just 3 animals. We have 21 animals which showed similar trend. So this all confirms that the population that we have isolated based on our in vitro method has either enriched metastatic cells with the cancer stem cell property, or it facilitates the upregulation of the cancer stem cells. So now we want to ask the question, what is the connection between the metabolic reprogramming or the altered metabolism and this cancer stem cell phenotype? So here we compared 3 different subsets. This is the parental monolayer, and this is the EMT phenotype or the spheroids that we developed. And this is the reversal, which is a reversal of this spheroid, which means we should have more metastatic or cancer stem cells. When we checked for the glycolytic pathway, my major interest is glycolysis. So when we checked for the glycolytic pathway, all these glycolytic enzymes, the red bar cylinder represents this EMT phenotype, and the green one refers to this reverse spheroid monolayer, and the blue one refers to the parental monolayer. You can see an upregulation of the glycolytic enzymes in all these major enzymes, hexokinase 2, GAPDH, LDH, and PDK. They are upregulated in the EMT phenotype, meaning here the glycolysis is the primary metabolic pathway for energy production and their survival. And when you reverse them, they tend to go down. Even though they tend to go down, they still remain higher than their parental population. But the point here is the metabolic reprogramming is evident at the early stage of metastasis. This is the early stage, and as you remember, this is the first stage, the epithelial mesenchymal transition. So the metabolic reprogramming doesn't come at the later stage or at the metastatic process, but it begins where the metastatic process is initiated by the EMT stage itself. So having seen the glycolytic pathways upregulated during EMT, we wanted to investigate what is the fate of cancer stem cell markers. Same way, they almost mimic their pattern, and you can see most of the EMT or the CSE markers are upregulated in the EMT phenotype, and once you reverse them, they actually tend to go down, but still they are higher than their original parental population, except the CDH1. We know now there are several forms of CDH1 which can complement for the function, but the take-home message here is during the EMT, not only the metabolic phenotype is altered, but the cancer stem cell markers also get induced during EMT, and that could be the reason why in the late metastatic cells we see upregulation of cancer stem cell markers and metabolic reprogramming. So with this, what we know is cancer stemness is involved in the manifestation of the phenotype, which is the cancer stem cell phenotype, and also tumor initiation and resistance and the achievement of the metastatic status. But how they achieve is probably, right now it's preliminary to say, probably through the metabolic reprogramming, because we have only one piece of data which shows the link between metabolic reprogramming and the metastasis, where you see the increase in the cancer stemness. So it's fair to say during metastasis process, the role of cancer stemness and metabolic alteration is critical. In other words, they mutually cooperate, and then they play a major role in the development of metastatic phenotype or aggressive phenotype. But what we don't know right now is which one is controlling what one, or which one, or is there any common regulatory pathway that activates both of them. What is the clinical relevance? So there are reports which show, like, if you target cancer stem cells by any combination strategy, it is possible you can completely eradicate tumor. So in a recent review article, it has been even hypothesized that conventional therapy kills bulk of the proliferating cells, but not the cancer stem cells. Whereas if you combine a glycolytic inhibitor, since we know glycolysis is upregulated, if you combine a glycolytic inhibitor, there is a possibility that you can eradicate tumor completely, because during the process of metastasis, the glycolytic process is upregulated, and you are targeting on one hand energy metabolism, on the other hand with your conventional therapy you are targeting the proliferating cells. So the summary here is the anchorage independent growth, which is EMT, involves upregulation of both glycolytic phenotype and the CSE markers, and the metastatic phenotype integrates by all means the CSE-like properties and the metabolic reprogramming. And finally, if you target energy metabolism in combination with your conventional therapy, you may improve the therapeutic outcome because you are actually targeting cancer stem cells as well. And these are the postdoctoral fellows who worked in my laboratory, and Swati was a master's student, now she joined for PhD, and I thank the Department of Defense and the SIR for their support. And thank you all. I take any questions.
Any questions from the floor?
Yes.
Did you directly measure any metabolites? I see a lot of quantitative RT-PCR, but no direct measurements of metabolites.
No, we haven't done it. So there are several projects or side projects that are in progress. So we have recently established a metabolomic facility which can quantify the metabolites that are altered during the process of EMT as well as during the reversal process. So it's in progress. We haven't got any data so far.
Are aware that there are conflicting observations in the literature that actually attribute an increase in ATP generation and oxidative phosphorylation to metastasis, in contrast to the Warburg effect in the primary tumor. So it is not clear, but I do understand and appreciate that there are publications along the lines of what you've just presented, but it's by far not resolved. It would have been nice to have direct measurements of ATP or direct measurements of metabolites in order to really pin down that they're dealing with a Warburg-like effect or you're just having the glycolytic pathway feeding into the oxidative
True.
phosphorylation. Because some of your marker genes don't interfere upstream.
True. So that is where the role of the microenvironment, Lisanti's group is doing excellent work there. So that is where the synergistic interaction between the fibroblasts which are surrounding the tumor and the actual tumor, they exchange or mutually benefit. The fibroblasts, they donate a lactic acid, and the tumors can take the lactic acid and use TCA. So even if you measure in a realistic condition the ATP level or any metabolite, we have to find a way to ascertain or confirm those are originally from the cancer cells, not by the surrounding microenvironment. It's still, arguable. I agree. So
You had a monoculture system, right? These were just the individual tumors.
True. Yeah, here it should be very straightforward. But then the argument could be: is it real or applicable to in vivo system? Because whatever we find, then it's an in vitro model, and whether it will hold up to the in vivo system. If not, then we have to go for other explanations, other mechanisms involved in it.
Yeah, as of now, I'm just from the data that you presented, I think it's not excluded that this is not
Possible. Yeah, I agree. Yeah, true.
All right, well, thank you. In the interest of